Well, hello, Bio 121. We're looking at Chapter 16 and gene expression. And in many ways, today is the day that we begin to actually answer that question that Markel asked, seemed like a lifetime ago. How does Mother Nature actually create the variation that's seen inside every single organism? And at least mechanistically, we still don't understand all of the pieces involved, but I can begin inside Chapter 16 to help you understand how it is that if you're made up of trillions of cells, let's go here. If you're made up of trillions of cells, this is you, right? How is it that the cells maybe down in your big toe are different than the cells that are on your earlobe? And then how did your body know when and how to make each of these? And again, in previous chapter 15, remember I said that some of the material when you're reading it is going to sound like we're simply repeating ourselves again and again. And the mechanisms are generally the same. But what we're doing now is we're asking if Mother Nature has 23 pair of chromosomes, just go CHR, chromosomes. And on those 23 pair of chromosomes, you have, your textbook says 25,000 genes in those 23 pair of chromosomes. How is it you can go from 20 to 25,000 genes to more than, 30,000 or so, let's go with 30K, proteins? And the number, of course, can actually be much higher than that inside any one cell. And the reason for that is what Mother Nature can do is she can regulate gene expression by using some of those same mechanisms we saw inside Chapter 15. And what I want to do is I want to just simply explain to you how it might work and what it might look like, because I don't think we're going to spend a lot of time describing, aside from alternative splicing mechanisms and then thinking about what that might be. I don't want you trying to memorize what's actually happening inside each distinct cell type. Because, to be completely honest with you, in the last 2 years, we've finally gotten down to single-cell gene expression profiles, and people still aren't certain, one, how that works inside of one cell, but two, what that means when cells are possibly adjacent to one another. So, what's actually going on here? Each somatic cell contains that full genome, okay? And that means you have to regulate the efficiency of it so that you don't waste energy. There's only so much space cells can maintain. Remember, this is a ratio of surface area. So you actually have to make sure that cell is doing exactly what it's supposed to be doing. And remember, each cell has a specific cell cycle, and that means everything needs to be done inside of that time frame, and it's coordinated. Most cells, barring some disease state, they all work in a coordinated manner, but they're not moving identically in time. They're moving generally in time, or relativistically in time. And this is actually how you end up building the different types of cell types that are required for, if this is an eyeball here, and inside this yellow piece here, that is the actual retina, and outside of that is the vascular tunic, and outside here is the sclera. These are all different pieces of tissue that are required, which means the cells that made them had different instructions to build that structure. Likewise, if we think about, say, here, a liver, a gallbladder, which is just underneath here, here's a pancreas, and here's a small intestine. Number one, how did Mother Nature know to make this, or make this ductal structure that gets the contents of your gallbladder now to your small intestine? And then how does it know to regulate the activities between the pancreas and the gallbladder such that you can offset the pH of the acidic materials coming into your small intestine? So differential gene expression patterns arise, and this has been the most active field of research. I mean, this is catapulted in terms of data analysis in the last 15 years or 20 years. What we can understand happens inside of one cell, second to second, okay? But those are static images. We have to sort of now build those static images into what happens inside of a real-life model. So inside prokaryotic transcription and translation. Remember, because you have a single circular nuclear structure with no nuclear envelope, you create these polyribosome structures, and regulation occurs at the transcription level. In eukaryotic cells, this is hopefully what you walked away with from chapter 15 at least, is inside the nucleus. You have a pre-mRNA structure we needed to modify. We created that structure, then we brought it out to the ribosome, small subunit, large subunit, brought in some tRNA. We started making now proteins from them. And here what happens is gene regulation can be regulated at a transcriptional level, a translational level, as well as post-translational modifications of proteins. So not only can we make different transcribed structures, we can regulate the translation of them. And even after all of that has done, we can now make post-translational modifications on an ever-increasing but required number of differences. So here's a summary. You find this inside your textbook. Gene expression regulation differences, right? So when you're studying for your exam, this is a great place to come back and go, hey, how are they the same? How are they different? This is really important. And one thing I'd like to highlight to you here is gene expression for prokaryotes is primarily at the transcriptional level. Whereas notice what we're doing now is we're changing the vocabulary from the previous structure. Transcription, translational, post-translational modifications. We'll spend some time looking at epigenetic transcription, nuclear shuttling, post-translational modifications, post-transcriptional modifications, okay, as well as translational modifications. Okay. Now, this is actually, in concept, I mean, this is, this is a textbook right here. This is a textbook's worth of information. We don't expect you to walk out of this class understanding how all of this happens. I mean, the molecular basis of genetic expression is a textbook that's, I don't know, it's like 400 pages long, 500 pages long, sort of thing. So what we want you to walk away from it is how are prokaryotes behaving, where they use repressors, activators, and inducers to regulate what's happening inside of here. And to do that, we want you to think, well, remember, it is a circular genome, and the biochemical pathways can be organized into blocks. And those blocks are called operons. Operons regulate 3 molecular types, okay, or are regulated by 3 molecular types. And this should have a general feel of how we thought about, say, maybe enzymatic competitions, where you have repressors that will suppress transcription, activators that will increase transcription, and inducers, which may do either suppress or activate. And you can see how you can use these as a switch. That's ultimately what you want to take away from this. So in this first part of chapter 16, we'll introduce prokaryotic gene regulation. Okay. Here is the prototypic system that's been examined since the beginning of genetic regulation analysis, right? We're using the production of an amino acid, tryptophan. The trip operon, okay, is a repressor operon, or it has a repressor operon. And what this means is when tryptophan is present, the trip repressor binds to the operator. And that means we will not make tryptophan because the RNA synthesis is blocked. So what does this mean? Here is the genetic sequence. Promoter, operator, trip E, trip D, trip C, trip B, trip A. You don't need to worry what these things are. That's not what's important. Is if you have sufficient concentrations of tryptophan, so when tryptophan is plentiful, the 2 tryptophan molecules will bind to the repressor at the operator sequence. Okay, and this complex will physically block RNA polymerase. So sufficient levels of tryptophan, remember 2 molecules will bind, it will stop the transcription and the translation of all of the materials downstream of here. Okay, so that's pretty nifty. Okay. Now, the repressor operon, okay, so when tryptophan is absent, meaning we don't have enough, notice what actually happens to the repressor. It literally falls off of the operator, and RNA polymerase can now go on. These negative regulators are proteins that bind to the operator that silence trip expression. Okay, so now we'll make plenty of trip will bind here, it will come back here and shut the system off again. And you can see how this becomes a feedback mechanism. Presence or absence of the product, therefore, going back to what we learned back in, I think, chapter 6 or something like that, you can see how it begins to all fill together. Now, when we're looking at, say here, a different one, we're looking at LAC-Z, LAC-Y, and LAC-A. These are sugars, catabolite, activator protein, cap protein, which is an activator regulator. If the first was a repressor, this is an activator. What you're going to see here is glucose supplies become limited inside of the cell. That's because what we need to do is we need to figure out if we need something to happen, proteins that bind to the promoter in order to activate gene expression. We recognize there's no glucose. We increase cyclic AMP levels. Remember, we took ATP, we convert this into cyclic AMP, and it binds to that cap protein. Cap protein here. Pay attention to the requirements. In the absence of cyclic AMP, cap does not bind the promoter. In the presence of cyclic AMP, cap binds the promoter and increases RNA polymerase activity. And now you can use this as a reporter system in the sense that once this happens, the RNA polymerase will come along, make LAC-Z, LAC-Y, and LAC-A, and you can demonstrate that this is functional. Supporting this idea now that you have an activator. But notice what's happening here. It's the presence or absence of glucose that starts this. It's not the presence or absence of the pathway itself, meaning what we saw with, say, our tryptophan system. Now, the LAC-OPERON also has this inducer operon as well. In the absence of lactose, the LAC repressor binds the operator. So if here's lactose, here was glucose. Make sure you pay attention to the differences in this, right? So glucose, we use cap and cyclic AMP. Lactose, we have a repressor system. And what's going to happen here is lactose now acts very much like, or I should say, looks similar to our tryptophan operator. Okay, so in the presence of lactose, the LAC repressor is released from the operator. Okay, so if we've made or we have sufficient lactose or we have insufficient, let's start here. In the absence of lactose, the LAC repressor binds the operator and transcription is blocked. In the presence of it, though, we're now going to allow for this to actually work. And you might think to yourself, why is Mother Nature doing this? It's because activation and repression depends on the local environment and the needs of the cell. So this is more of a nuanced, God, I hate using that word, a nuanced mechanism for actually regulating gene expression. So, how can this actually work? The LAC-OPERON itself has that cyclic AMP cap complex stimulus activity. However, even in the presence of cyclic AMP, RNA synthesis can be blocked when a repressor system is actually bound. So here's what I want you to think of this like. This becomes a series of ifs and thens, and we won't be testing you on the ifs and the thens. We want to make sure you know what an inducer operon is, what an activator regulator is, or activator molecule, and then what a repressor operon is. But here's something to help you begin to see some of the complexity that is used to regulate gene expression in prokaryotes. Okay. Whoops. So we'll pause here before we actually move on to the eukaryotic system. Okay.